Now, the first group of research I'm going to talk about this is my thesis research from the University of Maryland. My thesis advisor was Dr. Jocelyn Ruggiero. And this work was done with the Institute for Systems Biology in Seattle, with Nitin Baliga and his postdoc, Kenya Whitehead, and also with the National Institute of Standards and Technology. Murad Disroglu there helped us out with a number of these experiments. Now, this is my thesis title, so of course it's very scary. It has all the wonderful scientific words in there that are designed to impress people and sound very scientific. What this means is that extremophile biology starts with trying to kill things. And if you can't kill it, you find out why you can't kill it. So what I did was I tried to expose them to ionizing radiation. Find out, when I stressed them, what did that look like? How did you prevent any damage from happening? And how did we repair any of the DNA damage that actually did occur in the cells? And the organism I used is called Halobacterium salinarum. And it was done with the National Research Council's first isolated it. So it's strain NRC1. First thing we found out, halophiles can survive a whole lot of stress. Turns out that you can dry them out and trap them inside of salt crystals. They're still alive just fine. Turns out you can expose them to vacuum, the same as low Earth orbit, which we did with the Goddard Space Flight Center. They survive just fine. And if you expose them to gamma radiation, as you see here, as you increase the amount of gamma radiation, survival, you can map on this chart. This is a logarithmic scale of survival. So here's marking 10% survival, and this is full survival. So halobacterium cells can survive up to about 5 kilograms. Now, to put that in comparison for you, E. coli are dead at about 0.1 kilogray, and you and I are even less hardy. Here we are at 0.005 kilogray. These things can take a lot of stress. So then I wanted to find out why. So it turns out when you stress an organism, you can group their response into 3 major categories. How do they avoid the damage? How do they respond to the damage? And then how do they repair the damage that they do have? So if you start with damage avoidance, my wonderful artistic rendering of a halophile cell with its flagellum there, cell membrane, cell interior, they actually have pigments. The pink pigments on their cell membrane work as sunscreen. It actually helps to mitigate the effects of high solar radiation. They also have phototaxis swimming towards lower energy orange light away from blue light, and they can also have aerotaxis, which moves them into the right position in the water column for the oxygen level. The salt inside their cells balances the salt outside the cells, so they trap all the salt inside their cells to balance things out. This is also the reason why these cells can never be taken out of a high salt environment. Everything inside their cells requires that salt being there. So if you put them into pure water, they burst open. So at the University of Maryland, we exposed these to gamma radiation. These are plates full of bacteria lined up around the gamma source. We wanted to see what would happen. So what we did was we were measuring things like DNA damage and protein damage. If you see the lines here, all it was different salt conditions. We wanted to measure different damages with different salt conditions. Same thing with the protein oxidation. Here, we're looking at different salt conditions and how much of these black spots we saw. Okay, what you need to take away from this, if you're not a molecular biologist? Translation, the salt inside the cells actually protects the cells. It turns out that when you put table salt inside the cells, sodium chloride, they'll kick the sodium out, they keep the chloride inside. That's the part they want. And it turns out that chloride inside the cell works as an antioxidant. It prevents the damage from ever occurring to the DNA and to the proteins. It's the first time we were able to actually confirm this inside a cell. Now, I wish I could tell you this would totally justify the amount of salt that I put on my popcorn. But unfortunately, our cells don't work that way. Our cells don't require the salt. So as much as I would love to justify my salt intake, sorry, this one isn't for us. The interesting thing that we also found was that the DNA can repair over time. What you're looking at here is pieces of DNA, intact DNA before radiation. At this time point, we expose them to radiation and then let the cells recover over 2, 4, 8, and 12 hours. You notice at 12 hours, it looks exactly the same as it did before we started. 12 hours, these cells can reverse all the damage to their DNA. And everything is in the perfect order and works perfectly the way it's supposed to. Now, trying to figure out how they pulled off that feat, we started doing analysis on their genome. This is a giant map. Every single dot on this map is a different gene inside the cells. These are the ones that we know what the gene codes for. All of this, we have no idea what that codes for. This is the daunting task that we face in the genomic era. We have a lot of information, and we need to start piecing our way through it a bit at a time. But if we take a look at one of these areas, what you'll notice is you see different colors. Well, the red in this case means there's more of that gene product after we expose these cells to radiation. They make more of it. They need more of that for some reason. If it's green, it means the cells shut it off after radiation. They wanted less of it. And so you can see that if there's pale pink, it was slightly more. If it's dark red, they needed a lot more of it. And this is a way for us to see what happens to these cells and which genes are important. And if we can start to identify which ones are important, we can start to figure out what each one of these genes is. Well, I showed you before that these cells can repair their DNA, but the interesting question then is how? Well, this is a model of how they think it may work in humans. If you've got a broken DNA end over here and the other half of your broken end over here, there's a huge protein complex here that tethers the two ends together and then helps to fill in this gap. Well, we're not exactly sure how this works in humans. So I was able to actually do some studies on these proteins, because remember, there are far fewer of these proteins in the archaea than there are in human. I was focusing on these two proteins, part of this tether that you see here. We were able to show some insight into which one of these was actually more important for cells. If you lost function in this one, it turned out to be more important than the RAD50. So that gives people an idea of where to look. This is important in humans because, again, these are the genes for breast cancer. They're involved with this pathway. So if we can start piece by piece to figure out all the genes that are involved, we can start to increase the complexity in humans and start to understand some of these disorders that can lead to cancers. So what other extreme environments are there in the solar system? Well, I'll tell you one thing, we found areas of high pressure. This is work that's been done with money from the Keck Foundation, and PIs for this were Marilyn Fogel and Andrew Steele. So just to give you an idea of what we're looking at, these are the moons around Jupiter. So here's the different moons up top. You can see a closer look at their surface and a high resolution magnification of that surface. You'll see these cracks. These are covered in ice. And the cracks you're seeing are cracks through the ice layer. And what's thought is that there's a liquid ocean underneath this ice layer. Question is, how thick is that ice? And, what's the temperature in that water? And nobody knows. NASA and ESA have just combined forces to do a mission to the icy moons. So this is going to be upcoming. But the issue here that we need to start to address is if life's in this ocean underneath, well, now you've got ice pushing down on top. You're going to create a high pressure environment. The one thing they do think they know for sure is that this would be a sailing ocean underneath. Another interesting place to look at is around Saturn. One of the moons around Saturn is Enceladus. And it has this giant plume. It's a geyser exploding out into space. And then what's thought was that it's actually water vapor and ice particles being shot up through a vent through the ice layer at the surface. And there could be possible hydrothermal activity underneath, which is what's pushing it up in this geyser.